My research program in the Department of Biological Sciences at the University of Manitoba aims to obtain a better understanding on how does the human brain grow during fetal development, and especially how the human brain is so different from other species. So in order for the human brain to grow properly, the neural stem cells residing in the fetal brain must be precisely regulated by different signaling molecules. So the projects in my lab take a very fresh perspective by looking at human brain development from the perspective of the placental contribution. And hopefully, we will gain a better understanding how do these signal molecules secreted by the placenta travel to the fetal human brain and regulate the neural stem cell there, and if any of these molecular and cellular mechanisms actually contributed to the human cognition. Currently, my lab focuses on a particular placental regulator, which is called growth differentiation factor 15. And we have generated genetically engineered mouse models to remove this placental factor from the mouse placenta and see if the brains will develop differently, especially the neural stem cell and neuroprogenitor cell biology and behaviors in the embryonic mouse brains. By focusing on the placental contribution to the fetal human brain development, we can also apply that knowledge to understand if placental dysfunction could lead to the onset of certain neurodevelopmental or psychiatric disorders, especially autism spectrum disorder. I have a very collaborative and friendly research team, including 2 master's students and 4 undergrad students who work together very well. This inclusive environment is especially important for all of our research achievement that we have. With the support of Research Manitoba grant, we can complement our genetically engineered mouse models by establishing a more physiological relevant model system to understand more about the human brain. Due to the apparent species difference between mouse and human, the unique features and characteristics that's human-specific will not be modeled very well by using the mice. We're planning to establish a placenta brain organoid co-culture model system. Organoids are these miniaturized and simplified organs that we can grow in the lab. They are these 3D culture model systems that can mimic the structural, functional, and biological complexity of a given organ. So by placing 2 different types of organoid, placenta and brain, in the same culture dish in our lab, we hope to better understand the communication between these 2 organs during development. And hopefully, the knowledge generated by using this very novel model system will provide us more insight on a better understanding of the human brain development and also some of the mechanisms involved. In Manitoba, there is an increased prevalence and patient rate for autism in recent years. And with the placenta brain organoid co-culture system, we hope to gain more insight in the molecular mechanisms leading to the onset of autism spectrum disorder. And ultimately, we hope to contribute to better diagnosis for autism and also maybe better intervention and more suitable training programs for all Manitobans.